Hey guys, my name is Abhishek and welcome back to MBBS Treasure channel. Today we are going to discuss the second part of type 2 hypersensitivity reaction, that is cytotoxic reaction. If you haven't watched my part 1 of cytotoxic reaction, that is type 2 hypersensitivity, please go to my playlist of pathophysiology and there you will find the part 1 of cytotoxic reaction or hypersensitivity. Okay, so let's begin our today's topic. In this video, we are going to discuss about the actual mechanisms through which the cytotoxicity is occurring, means the cell lysis is occurring, and we know from my previous video that it is mediated by antibodies like IgG and IgM. To recap the previous knowledge, I want to say something about the first part of my video, that how this reaction was occurring. This reaction or this type of hypersensitivity, as we know, it's a immunological disorder, and this disorder mainly or primarily occur in the primary lymphoid organs. And as the primary lymphoid organs get disordered, so there will be increasing in the amount of antibody due to increasing in the amount of B lymphocytes, and that leads to the presence of increased amount of antibodies freely present in the blood vessels. Okay, so when they are freely present, so they will be trying to find the antigens, whether it is harmful or not. If it is harmful, then it is obviously good for our body, but if the antigens are not harmful, then what will be the problem? The problem will be like it will be autoallergy disorder. Okay, so next comes when the antibody will bind with the allergens that are present over the surface of the cell membrane of a cell. Then what will happen? It will be doing some kind of mechanisms through which there will be dysfunction and destruction of the cell, and that is nothing but the cytotoxic effect of reaction. Okay, so that we are going to see that what is that mechanism of action through which there will be cytotoxic effect or this cytolysis. Okay, so mainly these mechanisms occur in three processes or three mechanisms, that are complement mediated cytotoxicity, first one. Second one is antibody mediated or opsonin mediated opsonization and phagocytosis. Third one is cell mediated cytotoxicity or otherwise known as Adccc. I want to make one thing clear, that it is antigen antibody interaction. So this whole process of mechanisms will be triggered by this antigen antibody complex, as this antigen antibody complex will trigger certain kind of factors releasing and functioning these mechanisms. So let's see first the complement mediated cytotoxicity. What happens? This is an example of RBC cell. Okay, and on the surface of it there are the antigens present, and to antigens the antibody IgG is bound. Okay, so what happens when this condition arises, when this complex structure is present inside, then there will be activation of some components in the blood plasma. What are these components? These are nothing but the complement components, or these complement components will be mediating the cytotoxicity reaction. What happens? This complement proteins, what do I mean by complement proteins? These are the group of small proteins that are present in the plasma and responsible for performing different kinds of immunological reactions, as there will be in case of infections, when there will be presence of some microorganisms and when the antibody will be attached to it. So to protect our body, this complement usually get activated. But in this condition, as there is a case of hypersensitivity and immunological disorder, so this complex will trigger the activation of the complement system. And how the complement system get activated? First, there will be the C1 will be getting activated, then C2, then C3, C4, C5 will be getting activated, and then after C6, C7, C8 and C9 will be activated. What happens in this complement mediated cytotoxicity? How this complement helps, or how this complement will proceed in the cytotoxicity reaction? What happens? This C1 activates the C2, then C2 activates the C3, C4, C5. Then after activations, there will be a process taking place, that is cleavage, and that is by some cleavaging enzymes. They will try to break the components of this C3, C4, C5 proteins into two halves, and first half will be called as A half and second will be called as B half. So after splitting the components of C3, C4, C5, what will happen? This C3A, C4A and C5A molecules will be produced. Okay, so these molecules like C3A, C4A, C5A molecules will act as a chemotactic factor. What do you mean by chemotactic factor? It means it will attract a chemical or a cell for the process of phagocytosis. Okay, and these cells are like neutrophils, so they are going to attract the neutrophils for the process of phagocytosis. Then if the complement systems or this complement will be fully activated means after this C3, C4, C5 activation, if this complex will activate C6, C7, C8 and C9, then with the C5b component and C6, C7, C8, C9 with more number of C9 component will make a membrane attack complex. Okay, so this will be formed as known as membrane attack complex. There will be a structure like this. Okay, there will be a structure like this. This represents C5b, C6, C7, C8, and there will be whole lot of C9 structures. Okay, so C5b, C6, C7, C8 and whole lot of C9 proteinaceous part. So this complement, that is membrane attack complex, will be affecting the cell membrane. How it will affect? It will be producing this mediators that is known as perforins. What is the function of perforins? Perforins will try to perforate the cell membrane. They will try to perforate the cell membrane. They will try to make a hole in the cell membrane. Okay, so when there is a hole on the cell membrane, then due to the process of osmosis, more amount of water will enter into the cell, and as the more amount of water will enter into the cell, what will happen? The inflammations, the cell will try to overflow and it will get rupture. Okay, so by that process the perforins does its action. Next comes the antibody mediated or opsonin mediated opsonization and phagocytosis. Here comes the special thing that, as we know, there are two types of fragments present in the antibodies. One is FAB fragment and other one is FC fragment. FAB fragment attaches with the antigen and FC fragment is free. Okay, so this fragment is free. In this condition, the FC fragment is bound with the C1 complex or the complement receptors, and here what will happen? These receptors are also free. The FC receptors are also free. So what will happen? The antibody mediated or opsonin mediated opsonization and phagocytosis occurs. It means there will be the normal process of antibody antigen interaction, first thing. Second thing, so there will be also presence of C3b component. What do you mean by this C3b? After the cleavage process, this part, okay, this part will try to move and try to deposit on the surface of the cell membrane. Okay, when they will try to deposit on the cell membrane, they will act as an opsonin. Second thing, so first thing was this antigen antibody interaction will act as opsonin, and the second thing, the C3b will also act as an opsonin. What do you mean by this opsonin? Opsonin is a substance that will be more susceptible for phagocytosis. It will try to increase the process of phagocytosis as it is more susceptible for the process. So as the antigen antibody complex present over the surface of the cell membrane and the C3b will be also adding on the surface of the cell membrane, this process is known as opsonization. So when the cell will be get opsonized, when the RBC cell will be getting opsonized, this will be attracting the macrophage, because the macrophage also have special receptor for the C3b component and also special receptor, that is Fc component of the antibody. As we know, this antibody is IgG, so it will be having special Fc receptor for the antibody. Then macrophage will perform its actions by the process of phagocytosis. So now we understood that opsonization will be associated along with this complement factors through these two opsonin conditions, and then finally the macrophage will come and perform the process of phagocytosis. This process of opsonization and phagocytosis will be most effective if the C3b and antibody opsonized the cells. If the both component C3b and antibody as an opsonin will be present, then it will be both very effective. The process of cell lysis will be very effective. Next comes the cell mediated cytotoxicity. As from the word, you know, the cell, different kinds of cytotoxic cell will mediate the process of cytolysis. And what are these cells? The cells can be natural killer cells, macrophages, eosinophils, and these cells have special Fc receptor. These cells have special Fc receptors on their cell membrane, due to which they can recognize the Fc component of the antibody IgG. Okay, so that is the reason why they will try to attach or they will try to bind with the antigen antibody complex through this Fc compartment. So that is the reason why it is known as antigen dependent cell mediated cytotoxicity, because here the antigen dependent means the antigen will say that yes, I am responsible for the cell mediated cytotoxicity. It will be attracting the receptors that are present on the cytotoxic cells, and the cytotoxic cells will perform the cytotoxic reactions. So that is known as ADCC, antigen dependent cell mediated cytotoxic reaction. So this whole process will be going to regulate the process of cytotoxicity. How? Because they will try to produce certain kinds of mediators. As in case of these macrophages, they will try to produce these kinds of mediators. What are these mediators? The mediators can be like perforin in the first phase. It can be like perforin. It can be like tissue necrotizing factor. It can be like lysosomal enzymes in the macrophages. And it can also stimulate the production of the oxygen radicals. And as we know, these are the factors that is responsible for the process of cell dying, and this cell dying will be performed by this apoptosis mechanisms. So in this way we are concluding that the cell is finally going to destructed by these cell mediators. Okay, so these are the examples that occurs in this whole process. Okay, so this was all about the cytotoxic type of hypersensitivity. If you completely understood what do you mean by cytotoxicity reactions and what are its mechanisms, please like, share and subscribe to my channel. Thank you so much for watching my video.